the histology of the tooth, now as reference, we must be able to locate first the central portion of okay, so as stated, we must be able to locate first as reference the central portion of the tooth, which is composed actually of loose connective tissue with blood vessels. That is fine. Yan. Kulay yung areas na yan. Those are actually the red cells inside a vein. So, aside from the veins, there should also be arteries and nerves in that loose connective tissue. That central portion of the tooth is called the pulp. The pulp cavity. Now, immediately external to the pulp cavity is a harder, calcified type of tissue which is called dentine. Now, okay, be oriented with the location and the span of the dentine. It completely encloses the pulp cavity. Now, ideally, above that should be the gingival line, or that should be the gingival line. Above the line of the gingiva, the crown of the tooth should be covered by a much harder, calcified connective tissue, which is called the enamel. Now, unfortunately, in majority of the slides, the enamel is not shown. So it should be external to the dentine, above the gum line. Now, the gums are lined by what type of epithelium?
Stratified squamous.
Okay, stratified squamous. And then there are portions of the gums, those portions which are subjected to friction with the food that is being chewed is covered with keratin. So mayroong portions ng gums natin yan which are keratinized. Okay, so that should be the gum line. Pababa. So that should be the dentine. Now, covering the dentine at the level of the gum line and then upwards, there should be enamel. As mentioned a while ago, almost all slides for the tooth will not show the enamel. Okay, let's go downwards. Now, if the upper portion is called the crown, how do you call the lower portion of the tooth?
Root.
Okay, the root. And then covering the root of the gum line should be the cementum. Unfortunately also, just like the enamel, the cementum is not yet developed in this tooth. It should be seen external to the dentine. Now, to give you an idea of the histologic appearance of the cementum, it should look like this. How do you call this socket of bone where the tooth is embedded? The alveolar bone. So if that is the dentine, that's the alveolar bone. That should be also the appearance of the cementum, which should cover the dentine at the level of the root. Unfortunately, as mentioned a while ago, this is still a developing tooth. So hindi pa nag-form ang kanyang enamel. Now, evidence of its immaturity is the presence of those columnar cells. So that is the dentine. At the periphery of the dentine, there are columnar cells. These are actually the cementoblasts, which are responsible for the future production later of the cementum. And then another evidence of immaturity, that's the tissue composing the pulp cavity, that's the dentine. These are the columnar cells which are responsible for the synthesis of the matrix of the dentine. So how do you call these cells? The odontoblast. And then, that's the alveolar bone. You should be able to see what type of cells in the lacunae. The 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 osteocytes. Okay, close up view. So the spaces are called, okay, lacunae, and then centrally located are the nuclei of the osteocytes. So that's the pulp cavity. That should be the dentine. Covering the dentine externally at the level of the roots should be the cementum. However, it is not yet formed. And then that's the alveolar bone. Connecting the alveolar bone to the cementum is the periodontal ligament. It is actually composed of collagen fibers. So, eto, immature pa lang, wala pa tayong makitang masyadong collagen fibers. However, as this tooth matures, this will be filled with collagen fibers to anchor the tooth at its socket. So that portion of, okay, that's the dentine, the gums, and then that portion underneath the gums is a minor salivary gland. Mucus or serous? Mucus or serous secreting salivary glands? Mucus. And clear mucin vacuoles at the apical portion of the cytoplasm of the acinar cells. So anyway, those are the things which you should be able to identify for this slide. So the pulp cavity containing the loose connective tissue, dentine, and then the gingiva or the gums, and then it should be covered with enamel on the crown, and then below the gingiva line at the level of the root, the cementum, and then that should be the alveolar bone, and then present here should be the periodontal ligament. So those are the things which you should be able to identify for that slide. Now, for the blood cells. Okay. Now, that should be the appearance of a mature red blood cell. What is the shape of the mature red blood cell? Okay, the biconcave. So that biconcave shape can be seen on the lateral view of the red blood cells. So parang ganyan siya pag naka-lateral view. However, on top view, that will be the actual appearance of the red cells. There will be a central area of pallor or central pale area. So that pale area is imparted by the concave or the biconcave configuration of the red cells. So the pink cytoplasm is imparted by what protein? Hemoglobin. Now, aside from the red cells, we are able to see two types of mature white blood cells in that portion of the slide. So here we are seeing a larger white blood cell with lobulations or segmentations in its nucleus. So ideally also, there are granules present okay? So there are granules present in the cytoplasm of that white blood cell. So those granules are actually now the primary and the specific granules. However, because of a more or less neutral color of the cytoplasm of that cell, and then the characteristic segmentations of its nucleus, that cell is called, okay, the segmented neutrophil or immature neutrophil. So because the nucleus is already lobulated. How do you call that immediate precursor of the segmenters we're in?
Bond.
Okay, bond cell. Now, let's say neutrophil, and then notice here that this cell is showing us a round shaped nucleus, which is more or less similar in size with that of the immature red blood cell. So, if that is the volume of the cytoplasm of that white blood cell, what can you say about the volume of the cytoplasm of that white blood cell? Okay, the volume of cytoplasm is very minimal. We can hardly appreciate the cytoplasm of that white blood cell. Now, so it has a round to oval nucleus with very scant amount of cytoplasm. What cell is that? Okay, that's a lymphocyte. So a lymphocyte will have a round-shaped nucleus with very scant cytoplasm. A neutrophil will have segmented or lobulated nucleus with granules in its cytoplasm with a much larger size compared with that of the red blood cells and the lymphocytes. So we have already described two mature blood cells. Identify that cell. Okay, that's a segmented neutrophil. How about that cell?
It's immature.
Okay, immature red blood cell. Okay, if that is a segmented neutrophil, that is a red blood cell, what cell is that now? A lymphocyte with large round to oval nucleus and with very scant volume of cytoplasm. Now, that is a mature neutrophil. This one here is an eosinophil. Now, let us characterize the features of the eosinophil. Now, take a look at the nucleus of the neutrophils. You will be able to see several lobules. On the average, a mature neutrophil should have 3 to 5 segmentations in its nucleus. Now, in the case of the eosinophil, they will only have 2 lobulations or 2 segmentations in their nuclei. Then what can you say about the color of the cytoplasm of that cell in comparison with the cytoplasm of that cell? Darker, and then actually, in ideally stained, okay, those granules, those are actually granules, they should have a bright orange appearance. So, bright orange granules, large, coarse, bright orange granules in the cytoplasm of a bilobed white blood cell, it should be an eosinophil. already identified a lymphocyte, a red blood cell, a neutrophil, and an eosinophil. The last mature circulating blood cell that we have to identify is a basophil. Now, what are the characteristic identifying features of the basophils? Just like the eosinophils, they will have bilobulation only of their nuclei. So, dalawang lobes na makita sa kanilang nucleus. However, we will not be able to see the outline of the nucleus of the basophils because the cytoplasm of the basophils will be filled with large, coarse, basophilic or violet-colored granules. So, yan ang characteristic description ng mga basophils. The nucleus, the outline of the nucleus will be completely covered by large violet-colored granules. So, halos makover na yung outline ng nucleus and then sometimes parang nag-protrude pa sa plasma membrane ang mga granules. So, that's the characteristic. Unfortunately, that is not the characteristic appearance of a basophil kasi maraming details ang hindi na-project ng ating apparatus. So, just remember, in order for you to identify a basophil, you will not be able to appreciate anymore the outline of the nucleus. The nucleus will be completely covered by large violet-colored granules, and then sometimes the granules will be seen protruding from the surface of the plasma membrane of a basophil. So that is how to identify a basophil. So now for the last cellular component of the blood, the last component, which is actually not a complete cell, but fragments of a large cell in the bone marrow, which are responsible for the formation of clots, the platelets. Now, how will you be able to identify the platelets? Those are the red cells, those are the white blood cells, and then those two irregularly shaped fragments are the platelets. Now, identifying features of the platelets is that they are very small. They do not show a definite shape. So, kasi fragments lang sila, and then they will have central area of granules. So, their peripheral portion will be lighter than their central portion because their granules are located at the central portion of their cytoplasm. We will not be able to appreciate any nucleus in the platelets. Now, we have actually missed one cell in the peripheral blood, which is actually the largest among all cells in the blood, the monocytes. Let us look for a monocyte. Now, for those who are not medical technologists, pag may mag-study kayo ng mga white blood cells later, so examine first the gross appearance of your smear. Now, you should be examining the tail end of the smear. Ang tinatawag na feather portion of the smear. Kasi, yung mga white blood cells are much larger than the red cells. Pag spread ng mga cells, madadrag at the tail of the smear yung white blood cells. Kasi nga, malalaki sila. So in that portion, you will be able to see more number of white blood cells than the other portions of the slide. Now, in a peripheral smear, the first white blood cell that you should be able to see immediately will be the neutrophils, because they are the most numerous type of WBCs in the peripheral circulation. And then after the neutrophils, you will be able to find next the lymphocytes. So sila yung second next na pinakamadami. And then the third most numerous cell type should be the neutrophils. The monocytes. Unfortunately, for this blood, for this smear, konti lang masyadong kanyang mga white blood cells. So how do you call that cell? Okay, that's a band neutrophil. Now, to appreciate better this white blood cell, by the way, we will be able to identify the white blood cells and differentiate them from the red blood cells because the red blood cells, when mature, do not possess nucleus. Mga white blood cells, they will possess nucleus. And then, take a look at the size of the red cells. And then compare the size of the red cells to the size of that nucleated white blood cell. So that white blood cell is much larger than the red blood cells. Actually, this type of WBC is the largest among all the cell types, and then it will have a characteristic indentation or a bean-shaped nucleus. So in ideally stained smear, the color of the cytoplasm should be blue-gray. So blue-gray, and then there should be indentation of the nucleus imparting a kidney shape or a vein-shaped appearance. So that's the appearance of a monocyte. Close-up view of the monocyte. So that's the size of a mature red blood cell, almost twice the size of the mature red cell, and then there is an indentation in the nucleus of the monocyte. Sometimes the indentation is too deep